This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 252, recorded on September 30th, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schechter.
Well, hello there. How are you?
Good to see you again. We missed you.
Yeah, well, here I am.
Did you miss us?
I sure miss you. Did I ever?
Good. We're glad. Also joining us from Washington, D.C., Michele Swanson.
Hello. I am in Grammy land. My daughter and her husband had a baby a month ago, so I'm here working remotely in their neighborhood.
Are you visiting President Biden now and then?
Not yet, but I am going to a rally on Saturday to support abortion justice.
Very good. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And today is International Podcast Day. How about that?
No. Really?
It is indeed.
It is.
Wow.
Now, you may ask, what does one do on International Podcast Day? Well, we make a podcast. That's
We
what
make a
Absolutely.
podcast.
It is an international celebration of the power of podcasts. It's just a marketing thing, I guess. Almost every day is a day of something, right?
Mm-hmm.
Michael, is there an International Microbiology Day?
We should make one. We should make one.
I bet there is one already. There's certainly a polio day.
There is a polio day.
Not too long ago. And, of course, today's the last day in September. Boy,
I don't know where the summer went. It
that went
just
quickly.
went by in a blink of an eye.
Well, before you know it, the pandemic will be over and we'll be in the end of 2022.
Promise? Do you promise?
I can't, unfortunately. I would hope. That's what I hope.
Yeah.
Yeah, we're still seeing between 2,000 cases per day in South Carolina. The trend is fortunately going down, but we're only beginning to see the beginning of football season, and it's pretty spooky.
Michele, are people masked up down in Washington?
They are. Certainly in this neighborhood, they are.
Good.
Yeah. I'm excited about these antivirals that we're going to hear about soon. I know you'll cover them on TWiM, won't you, Vincent?
Yeah, we will. I'm just hoping we don't immediately get resistance to them.
Right.
Because when you use any drug as a monotherapy for viruses, especially RNA viruses, you get resistance rather quickly, and they can't use them in combinations yet because they haven't been tested. So I hope they're used judiciously. But
each one has a different target, I hope?
I only know one has an RNA polymerase target, but hopefully, yes, that would be good.
A cocktail then.
Yeah,
with different targets.
All right, we have two papers for you today. And to start us off, Michele will tell us about something interesting.
Yes, this is colicin-mediated transport of DNA through the iron transporter FEP-A, and this was published in MBio in September. And it is from a group at Oxford, collaborating with colleagues at Johns Hopkins. And they are Cohen-Cott, Harmoliker, Pham, Webby, Housden, Elston, Hopper, Mohammed, Robinson, Gray, and Cleathouse. And the topic is, as the title indicated, how a antibacterial toxin called colicin actually gets across the formidable barrier of a gram-negative bacterium, which has an outer and an inner membrane, to get into the cytoplasm and cause damage. So it turns out only a single molecule of this toxin, colicin B, is sufficient to kill E. coli. But how do they do it? That's the question. To understand how colicin B gets into the microbial cell, they do a simple experiment where they tag the colicin molecule with a fluorescent moiety so that they can just mix this tagged fluorescent toxin with E. coli, and then look at the cells under the microscope and ask, did it get in or not? So in figure 1, we can see in really discrete bright spots that colicin B can indeed get into wild-type E. coli, but if they mutate the E. coli and eliminate its known receptor, which is FEP-A, then the toxin can't get in. Or if instead they delete a small moiety on the toxin molecule, which is a box that binds to the transporter apparatus, the TON-B box, it still doesn't get in. So they've identified now on the toxin a TON-B box, and they've confirmed the receptor FEP-A, and they also have an assay now. They then do a nifty experiment where instead of putting a fluorescent molecule attached to the toxin, they add a short DNA molecule. And believe it or not, this can now ride in to the E. coli cell, piggybacking onto the toxin and get into the cytoplasm. So this is exciting, and we'll talk about it at the end because we can imagine a number of applications then of using this translocation system to introduce DNA or other cargo into gram-negative bacteria. So that's really the motivation for understanding how this works in detail. So to begin to look at that, they mix their colicin toxin with cells, allow it to bind, and then lyse the cells and purify the toxin bound to its receptor, and then use a technique called mass spectroscopy to define what is in that protein mix. And what they find is they're equal parts of the toxin and equal parts of the receptor. So they can deduce then that one toxin molecule binds to one receptor protein. So that's a one-to-one stoichiometry. They then want to look at what is the configuration of these two molecules when they're bound. So they've used some really neat methods here that include getting a three-dimensional picture of the translocation complex. So they do that by first going to a protein data bank. So this is an online resource that the community has been adding to whenever they solve the structure of a protein. So rather than going through really detailed work of determining the structure of your protein, you can first look in the protein data bank and see if that's already been done. So they were able to use this data bank to pull out the known structures for both the toxin and its receptor. Now, to deduce how the two might bind and what configuration, they use another public resource, which is called Rosetta Software. So this is software that's been developed by the community for molecular modeling studies, to look at docking of proteins on one another, and design experiments. And what's neat is that more than 150 people from 23 different universities all around the world have been adding to this Rosetta code to allow experimentalists now to go in and for free use this modeling software to sample in three-dimensional space many different configurations of the many possible configurations. What is the most likely binding architecture? So they did that, and they did get a nice 3D picture where they deduced that the toxin could bind to its receptor in a particular configuration. But of course, they wanted to verify what the modeling software had predicted. So to do that, they went back to just some classic biochemical technique, which was they used a chemical crosslinker, which is a small molecule that will go in and bind to particular amino acids. And then when you activate it with UV light, it will cross-link to whatever amino acid is close by. So when they do this method, they identified 3 different amino acids on the toxin that interacted with 3 different amino acids on the receptor. So this basically acted as 3 landmarks that they knew had to be close together in space, because when they did this cross-linking either on purified proteins in vitro or with whole bacterial cells that had been incubated with the toxin, they saw the same 3 cross-links. So 2 of the 3 of these cross-links made perfect sense when they looked at their Rosetta computational configuration 3D model, but one didn't. So that one that didn't was in the N-terminal tail of the toxin. So what they did is go back to the Rosetta software, and you can just change the constraints. They can say, okay, we want the N-terminal tail of the toxin to be able to flop around and explore different configurations. So when they did that, they were able to, the software generated what looked like a stable complex where the tail had now moved into a different position. So they verified that that binding could happen in real life by, again, going back to their cross-linking strategy and identified three-dimensional configuration for the receptor and toxin binding. What was interesting, though, is there was yet a third two more crosslinks that they detected when they incubated their toxin with whole cells that they couldn't place using these methods. So what they did next was use this Rosetta software to simulate what they know happens when the receptor normally delivers its normal cargo into the bacterial cell. So this receptor protein, FEP-A, is usually what E. coli uses to bind to an iron-binding protein or a siderophore and translocate that iron across the membrane into the cell that can use now that precious metal for its enzyme function. So they know that when FEP-B normally translocates its iron across, there is a part of FEP-B that swings out of the way to open up the translocation pore so that the iron-bound protein can get into the cell. So what they did is modeled letting that FEP-A arm swing out of place. And what they found is that a similar structure on the toxin then slipped in and took its place. So kind of like a bait and switch strategy where the E. coli FEP-A arm that is a TON-B binding site, when it slips out of the way, then a TON-B motif on the toxin slips into its place. So it has basically taken the place of what the normal cargo is, and now the TON-B system does its usual magic where it takes advantage of the proton motive force across the inner membrane and pulls that cargo into the cell. So this is a really elegant mechanism that they were able to deduce by using a combination of fluorescence microscopy, just looking at cells and seeing what molecules can get in and which genetic modifications block its access. And then using mass spec and this really neat Rosetta Stone software to do some simulations and modeling of 3D structures to deduce and then verify by chemical cross-linking experiments and genetic experiments the deduced structures. So to summarize, they have been able to find that this colicin B toxin is able to exploit the E. coli FEP-A receptor on the bacterial cell that will is normal cargo for this TON-B dependent translocation apparatus that pulls molecules into the E. coli cell using the proton motive force. And it does this by a neat bait and switch strategy where an arm of the toxin protein has a similar motif as the normal cargo. So when the normal cargo opens up the translocation pore, the toxin slips into that very space and gets into the E. coli cell. So having figured this out in molecular detail, they now have a better handle on what moieties are driving translocation, and then what amino acids and regions of the protein could be available to modify if they want to, or when they want to now develop this as a mechanism either to direct DNA into the cell, into gram-negative cells to say for genetic engineering purposes, or to attach small molecules that themselves could be toxic. So you can imagine that this could be used as a new strategy to design antibiotics that would specifically target cells that encode this TONB translocation system.
And since colicin is specific to the genus and species of the microbe you're attacking, you could really target and trim the microbiome without nuking all the good actors in a microbiome. And if this concept holds across gram negatives, colicin obviously is for E. coli, but if this holds across gram negatives in that they have similar transport devices in order to get these charged molecules into the cell where they could do useful things for us, it would be a leap forward in developing new anti-infectives that could be targeted, because right now we're mostly concerned about just scorching the microbiome with an antimicrobial therapy, not knowing how long it will take that microbiome to recover. And if it does recover, will it be with the right microbes? For example, we often suffer from C. difficile outbreaks because of antibiotic use.
And to your point, Michael, there's reason to be optimistic because the authors point out in their discussion that another toxin called piocin uses a TONB-dependent transporter to get into Pseudomonas aeruginosa, which also causes quite a lot of disease. So some of these moieties are broadly conserved, and there's reason to hope that this can be applied to a number of different bacteria.
Michele, how efficient is the DNA uptake? Is it better than, say, making cells competent and transforming them?
Yeah, they don't go into that detail. Their intent for that experiment was just to ask whether adding a molecule, some cargo, would interfere with the translocation mechanism.
Yeah.
But they don't go into the details of the energetics.
My guess is because DNA is highly charged, it would be one of the more challenging cargo moieties to truck in. So that's probably why they used it. They could easily make DNA, whereas it would be much harder to make some of these small molecules and proteins.
So this would have use for getting other things into cells, small molecules, proteins, maybe not DNA, right?
Yep, that's where they want to go with this, and it's quite a simple system.
I would bet electroporation is probably much better, where you use brute force in order to force the nucleic acid into the cell.
Yeah, which is great for experimentalists, but not so practical
not so much for
patients.
No, you don't want to weld the patient with electricity. No.
All right, a little mixture of basic science and applied, right?
Oh, yes.
With really neat demonstration of using these community-developed tools, the protein data bank, so that you can go and not have to solve the structure of a protein yourself. And then also this Rosetta software, which is constantly being upgraded by the community computational biologists for looking at 3D protein structure.
All right. Thank you, Michele.
My pleasure.
Now, Michael, can you give us a charge?
I can give you a charge. Now for something completely different. I think this paper is likely going to blow the minds of most microbiologists. And as we walk through the data, it would seem that this paper should be part of a narrative of one of those long dialogue scenes between two scientist type characters from an Isaac Asimov novel like Foundation, which is a very interesting television series that premiered last week on Apple TV. Not sponsored here, but we could always use the support, Mr. Cook. Shameless plug. Since this is International Podcast Day, I thought I'd sneak that one in. But seriously, the paper is entitled Silver Nanoparticles Boost Charge Extraction Efficiency in Shigella Microbial Fuel Cells. And it's from UCLA's Departments of Chemistry and Biochemistry, Material Science and Engineering, Bioengineering, and the Nanosystems Institute. And these are all at UCLA. And if there was ever a team science project, this clearly highlights how team science can really do some pretty cool things. The authors are Cao, Zhao, Peng, Xu, Ding, Song, Guan, Li, Wang, Zhu, Fu, Wang, Liu, Nielsen, Weiss, Duan, and Wang. And again, from our perspective, we need to be looking microbial from both the perspective of energy generation and carbon sequestrations. Some institutions are going there. For example, Vincent's institution, Columbia University, has pledged for the future that it will no longer install new fossil fuel connections in any new construction, refresh, or renovation projects. And to support this transition, the university will evaluate how to fully electrify the campus by replacing the on-site combustion of fossil fuels with clean, renewable energy sources. And microbial fuel cells happen to be one such solution, if and only if we can begin to generate gigawatts of energy from these remarkable devices. And as the title of the paper describes, what we are going to witness from the data presented is the in situ building of an electrical grid to efficiently extract energy from the bacterium Shigella. What is most remarkable is if you follow the build an electrical grid metaphor, you will appreciate the why and how the microbial fuel cell they describe is able to achieve the current densities, the power densities, and most importantly, the Coulombic efficiency rates that everyone in the community of electrochemical microbiologists is raving over. Namely, that Shigella and the silver microbial fuel cells delivered a maximum current density of 3.85 milliamps per square centimeter at a power density of 0.66 milliwatts per square centimeter and a single cell turnover frequency that was double anything that had been seen out there ever before. And it was 8.6 times 10 to the 5 turnover frequencies per second, which is all considerably higher than anything that we have seen to date. Now, to appreciate the data, I'm going to define some of the common electrical terms that most microbiologists like me have likely forgotten since their time spent on electricity and magnetism units of my college and high school chemistry and physics classes were in the last century.
Plus, many of our listeners haven't even had those classes.
That's
so it's
true.
Great that you are making this accessible to our broad audience here on Podcast Day.
So first, current is simply the number of electrons moving through a wire, or in this case, the bacterial membrane, and into the fuel cell or anode per unit time. So at its most basic, current is simply flow, and it's measured in amps or amperes. And an amp is an international unit for measuring current. In actuality, an amp is a measure of the quantity of electrons, or sometimes called electrical charge, flowing past a point in a circuit where you actually look down and you measure how many electrons are flowing past. And 1 amp means that 1 coulomb of electrons, which is 6.24 times 10 to the 18 electrons, are moving past that single point in that circuit per second. And so we're at milliamps. So we're moving 10 to the 15 electrons past this one point in time. So to think about it in something that I can put in my head easily, it's like measuring water flow. How many gallons pass a single point in a pipe in a minute, or gallons per minute? Now, power is the product of voltage and current, and it's simply the work done in moving the charge through a resistance, whether that be a pipe, whether it be the wire in your house, the bacterial membrane, or even running your cell phone. Voltage, if you will, is the pressure or flow rate from the electric circuit's power source that pushes the charged electrons or current through this conducting loop, whether it be a wire, whether it be from the membrane to the anode of the microbial fuel cell, enabling the current then to do work such as illuminating a light, or the way you always think, or the way I was originally think about power is the water wheel metaphor, where water turns a wheel that moves a stone, that grinds seeds into flour. And so you can effectively see how voltage would be involved in making things work. Now, Coulombic efficiency is really a math expression, and it's an indication of electrochemical reversibility. And as a practical measure, power and energy densities are calculated for how quickly you can get rid of the electron. Consider the electron as a hot potato. You want to take it in your hand and pass it on as quickly as possible. And although in the past, the energy efficiency was almost close to unity for all electrode materials that were used for everything from lithium-ion batteries that power our cell phones and computers to microbial fuel cells. And this factor is critically important as we begin to think about new high-density materials, since the energy density can be a way to get things to practical development. So with that physics/chemistry electrical review as prelude, now on to the paper. So microbial fuel cells in their simplest form simply are nothing more than reactors that will take a substrate and convert it into product. In the intermediate in the product, which is more bacterial cells, is effectively electrons. Electrons are the waste of metabolism. And the great challenge for all microbes, or all living cells for that matter, is where to dump your waste. So think back to Columbia University's challenge of a fossil fuel-free campus. What might they use to obtain electrons? Certainly there's the sun, and then there's the wind. You know, where they're going to get the wind is harvesting the energy from faculty meetings, all that hot air that comes out at faculty meetings. And finally, waste, most likely from the waste that's generated from dormitories, lavatories, and buildings, things like that. And most campuses are really a micro city, and they all pretty much have sewage treatment plants, which has a lot of carbon. Now, there are a diverse range of bacterial species that can effectively make use of these wide range of, if you will, biofood, the waste, and they can make microbial fuel cells an attractive technology for renewable bioelectricity production or power generation from biomass and wastewater treatment. Among the microbes that do this extremely well is this facultatively anaerobic gram-negative rod, Schmidt, most of which are found in extreme aquatic habitats where the temperature is very low and the pressures are very high. So this is a very hardy bacterium. And Schmidt, from most of our listeners' standpoint, are bacteria that are normal components of the surface flora of fish and are implicated in fish spoilage. Now, the authors point out in their introduction that the current density and power density obtained from typical Shigella microbial fill shells is generally too low for practical applications. Now, the issue is that at this low power output, recall that power is amps, that is, they ask to do work, so it's largely limited by the bacterial anode, where the microbe transfers its electrons to the fuel cell, and then the electrons are effectively what does the work. So the microbes need a more effective anode to dump their electrons, and that's the focus of this paper. They are effectively going to describe to us a very efficient anode that can literally take bacterial electrons in a thermodynamically efficient way. So if you think thermodynamically, it's the driving force. You want the anode to broadcast a signal, give me your energetic electrons. I can take all that you can deliver. So then the challenge is how to encourage the bacteria, in this case Shigella, to stick to the anode and establish a biofilm, burn the carbon, and transfer the electrons. Now, Shigella does not have conductive pili. In previous TWiMs, we talked about pili, which are protein structures that microbes have shuttled electrons down. Shigella doesn't have these conductive pili, but nevertheless, in this paper, they describe a rational strategy to boost transmembrane and extracellular electron transfer processes in the Shigella microbial fuel cells. And the way they did that is they used reduced graphene oxide silver nanoparticle scaffold in their anode construction. And in their beautiful studies, the systematic approach that they used is they used simple carbon paper or graphite, and then they used a graphene oxide, which is effectively a lattice of carbon and oxygen, and then they used a reduced graphene oxide in which silver ions were coupled. And the microbe then sends the electrons to the membrane hybrid that in turn releases a silver ion that the microbe then literally transforms that silver ion into a wire. And it literally attaches it to the microbial outer membrane. It's as if it's really analogous to you are hooking your own house up to an electrical grid. You are attaching a wire to the anode to send electrons to it. Now, to fabricate a functional microbial fuel cell, the bacteria must have carbon, and in this case, they fed it lactate, which is a reduced form of carbon. We are all familiar with lactate. It's lactic acid, if you will. And Shigella has the ability to form dense biofilms on electrodes if and only if they can offload their waste efficiently. And Shigella principally does this through the burning of this reduced carbon source, lactate, through its enzyme, lactate dehydrogenase. And we all can appreciate this enzyme, lactate dehydrogenase. We know it when we exercise as we experience the lactic acid burn by our muscles, secondary to our muscles running out of our primary electron sink, oxygen, to dump our waste electrons from our own metabolism as we require more work from our muscles during exercise. Here it's no different. And the measure of whether or not it's working is first whether the microbes can form a dense biofilm on this new surface they've constructed. And second is a measure of the current transferred as a function of time, which they can conveniently measure as volts. Then the current density is the number of electrons per unit area. And finally, the power density based on using, again, the three electrode materials that they are comparing to: the carbon paper, the reduced graphene oxide, and then the reduced graphene oxide doped with silver metal nanoparticles.
Michael, can you give us a sense for what the apparatus is? Is it just piece of paper immersed in broth, or
it is? It's as simple as that. It's literally flimsy, and it's a dual chamber system. And in the show notes and in their supplemental figures, which even if you're not a subscriber to Science Magazine, you can always download the supplemental figures for free. And that's where they have their beautiful illustrations. And so I'm hoping that Ray will put into our TWiM post the first figure from the supplement, namely how they envision the bacteria's moving the electrons from the inner membrane to this substrate. And it describes, the simple scheme of transferring electrons to the electron carriers in the membrane, ultimately going to a sink. And in this case, it's the anode or the carbon paper, the graphene oxide or the graphene oxide silver. And I know that many of you are going to say, I think silver is antibacterial. And again, their supplement, they show that, well, silver is indeed the antimicrobial in the case of E. coli, but in the case of Schmidt, it's not. Schmidt is one of those microbes that is refractory to many things, and it's very refractory to toxic metals. And Schmidt was not adversely affected by this. And they do some beautiful experiments in their supplemental work that actually illustrate that Schmidt is able to withstand the silver.
Yeah, they have a nice picture, Michael, in the sup data of their.
Yeah.
It's a plastic apparatus with two chambers and wires attached to it. The bacteria are inside. I suppose if they wanted to scale this up, they'd make big ones, right?
Yeah. Or they'd make mini ones, and then
Lots
they
of
would
them.
have the wires coming out, and they would run them in series or parallel, and all the tricks that we use for batteries. Remember, the way a Tesla runs is with little tiny batteries that you literally have in your computer. And they effectively have thousands of them literally in the battery pack, all wired together. So this is how I would envision Columbia University would effectively deal with generating electricity on the fly, is you would, make a physical plant that would have lots of these microbial fuel cells that you would be feeding waste materials to, capturing biomass, which would give you carbon sequestration as well.
And then you'd have to take the output of this cell and use it to charge a battery so you could store it, I guess?
Yes. Or you could use it in real time.
Yeah. I guess you could also run a car on it, but you'd still need batteries, right?
You'd still need batteries to hold, because, again, this is only generating milliamps.
Yeah.
And the challenge from my days with DOE, they always want to generate gigawatts.
Yeah.
And but now the authors show this in elegant detail. So the first set of experiments they show, and we're going to use some of the same techniques that Michele described. And it's really a very straightforward live dead staining technique that many of us have used in the lab. And they use confocal laser scanning microscopy and a Cyto 9 dye. And remember, this is live dead staining. So if you have a live cell, it'll bring the dye in and it will fluoresce green. And so the more green the image is, the more cells you have, the more alive they are. So the first question they ask is, how well are the microbes attaching to the various surfaces? And in their first figure, A, B, and C is carbon paper, graphene oxide, and then the graphene oxide with silver. And it's a green cloud.
They love silver.
It's a green cloud. I mean, and when you look at the scanning electron micrograph, you can see that the bacteria are beside themselves. They're literally stacked on top of one another. Talk about dense pack. And this is precisely what you want to see on a microbial fuel cell. Next up, as you might imagine, is they measure how the different electrodes perform from the perspective of generating energy. So they're feeding them all the same thing, lactate. And again, you see that the graphene silver is performing the best. It's making the most number of volts per unit time, and it plateaus at a nearly constant value. So it's producing constant voltage, and it basically reaches that point in about 2 days.
And the data are just beautiful. I mean, the difference between the silver versus the carbon paper are just it knocks your eyes out.
And it's not a fluke, because they actually do a refeeding experiment, which is in panel C of their second figure, and they refresh it. And what you see is when they run out of carbon, they stop making energy, and then you feed them again, and bang, the fuel cell goes back to where it was. It continues along until they consume the carbon. Then it crashes. Boom, they feed it again. And it's night and day.
Robust.
And the currents densities, again, you don't even have to understand the significance of what current density means, other than the fact it's how many volts are being generated per unit area. And the more volts per unit energy, the more efficient things are going to be. I mean, it was so incredible just to look at it. And when you look at the bottom panel of their second figure, you can see that they compare this graphene oxide silver fuel cell to published works. And they look at some of the published work that has been in the literature, and it's night and day.
And those published works are on different compositions, fuel cells that are made differently.
Yes.
And it's, wow, what a difference.
It's, things like polyaniline and graphite fiber brush, graphene platinum, graphene felt, and they're looking at steel. It's really, now this should not surprise anyone who has ever taken a chemical engineering class. But if you have more catalyst effectively present to burn the substrate, you should get more product. And that's effectively what they're seeing here. Right. And the product that they are measuring is not necessarily biomass, though that does come into play here in a minute. But they're measuring the current density. And it can be attributed to a larger number of bacteria, that green fog that we saw on their on their confocal scope, of the biofilm growing more efficiency. Or it could simply be a combination of the two, where the electrons are just more efficiently being shuttled out of the bacterium, because that has always been the bottleneck, how to get the electrons out. And what they show is that the silver nanoparticles are actually inserting themselves into the bacterial membrane. They're literally wiring the bacterium to the anode.
And this is absolutely stunning microscopy that they use.
I mean, today when we're recording this, the country is voting on the infrastructure bill. and these microbes are making infrastructure. They're making an electrical grid, which is real. Without any tools, they're just doing it simply by thermodynamic efficiencies. And
And just to emphasize that what he's saying is really well grounded in direct observation. The method that they used was to take the bacteria, the Shigella, and make what we call thin sections. So they're already quite small, but then they slice very thin sections and then look at them by scanning transmission electron microscopy in combination with an elemental microscopy.
X-ray diffraction.
X-ray diffraction, so they can separately visualize where the carbon atoms are and where the silver ions are. And that's how Michael was able to conclude that the silver is actually embedded directly in the membrane of the Shigella, which accounts for its efficiency.
In fact, so you mentioned the thin sections. We have some correspondence from the first author, Bo-Sheng Cao, who said, "My most impressive memory is from the ultratome, an apparatus needed to cut the embedded bacteria into 60 to 100 nanometer thick layers for EM analysis, as you've heard. Although difficult to operate, I finally made it after tons of practice. The ultra-thin sections are laying on the surface of the water, uniform and tidy. Because of their ultra-small thickness, they were just like small pieces of rainbows floating on the water. That was the most beautiful scene I have ever seen in my whole project."
It's amazing.
I mean, this is it's phenomenal, and the turnover frequency is a concept that many enzymologists are familiar with. It's how efficient an enzyme is, and remember, the enzyme has to go from substrate to product. And there's always a limiting component in that reaction chemistry, and it's to get rid of the waste. And the turnover frequency, and this is what's in their third figure, is actually double. And their turnover frequency is illustrating that the carbon paper and the reduced graphene oxide electrodes show a comparative turnover frequency at about 4 times 10 to the 5 per second, whereas the reduced graphene silver electrodes showed a turnover frequency at 8.6 times 10 to the 5 per second, which indicates a much higher electron extraction or transport efficiency to this particular anode. So up to now, we've talked principally about making a better microbial fuel cell, sort of an engineering thing. But as this is this week in microbiology, we need to ask ourselves how the microbe is doing this. Now, Michele sort of gave away the punchline, and the microbe is not dying from the silver ions. The way they phrased this experimental question was, how was the reduced graphene silver nanoparticle able to increase the turnover efficiency? And again, I encourage you to take a look at their figures. The thin section electron microscopy and the elemental analysis is beautiful. You see these little specks of silver in the rightmost panel, and fundamentally, it's effectively showing that they're putting in many doorways to dump their waste electrons, which then increases the efficiency. And they go through some stoichiometry in their discussion about how many molecules of lactate dehydrogenase are present in a cell. And they show, and this gets to the efficiency process. Now, if you consider there are 1,000 lactate dehydrogenase molecules per bacterium, the turnover frequency estimates for a single lactate dehydrogenase is about 394 per second for normal Shigella, but it's about 860 per second for Shigella silver hybrids. And so that compares favorable to those reported in the literature for Shigella. And they postulate then it's because these electrons are no longer bottlenecked. They have found a doorway out, and they can quickly get to the anode, and the microbes are just beating a path to this surface area to effectively charge it up. So that's basically it in a nutshell, is that the Schmidt biofilm is able to do this extremely well on this graphene silver matrix, and which leads me to ask the question, and why when I was reading the paper earlier this week and I saw about how Columbia announced that it was going green, I said, I wonder if they're going to think about a role for microbial catalysts in helping us all address climate change and our increased electrical needs.
What's the timeline here, Michael? When can we see this used?
I think it's an engineering issue. I think they just have to scale it. Schmidt, I think, is ready to go. We're not even making mutants. We're just pulling Schmidt. I mean, the Department of Energy has been looking at this. Derek Lovely has been working on this topic. There have been a number of labs. The UCLA group is one of the premier groups in the country.
Hemmer-Regera at Michigan State.
Yeah, that's right. There are so many out there that have been looking at this. And one of the cool papers I saw at ASM's Microbe this past June was about microbial cables. These are bacteria, principally Geobacter, that make these cables, and they're able to send electrons through a distance. But I think this offers more promise because the catalyst is directly linked to the anode. So the efficiencies and the power densities and the Coulombic efficiency is how quickly does the bacterium get rid of that electron, send it over the wall so it can burn another carbon atom.
And I see from the acknowledgments that they have submitted to the UCLA Patent Office an invention disclosure on this fabrication, and their work is funded by the Office of Naval Research. So let's go.
We have some information on the first author, Bosheng Cao. Bosheng was educated at Jilin University, where for a Bachelor of Science in Chemistry. That was back in 2016. Then actually got a BS in Business Management, and then went on to get a PhD in Material Chemistry at the University of California, Los Angeles. And Bosheng has done has had a lot of work experience over the years, working in labs of different sorts and at UCLA and so forth. When asked, what made you have a career in science, Bosheng said, well, I'm not really sure, but I have to say my two mentors, Professor Duan and Professor Huang, were really key. They always told me I should find the core essence of a scientific problem, and now says finding the core essence makes me happy as well as boosting the core essence. And I told you about the rainbows floating on the water. Bosheng loves to work out in the gym and loves to play the violin, two very different activities. He writes, both of them can make me feel relaxed, but in completely different ways. Maybe one way is through the mind and the other is through the body, I guess. And when asked for some advice for aspiring scientists, Bosheng wrote, well, from my own experience, always do your best, but relax your mind. This may sound self-contradictory, but it is what I learned on my way to the Doctor of Philosophy in Bold's philosophy. In scientific study, it's always important to work hard, but we can't expect that every experiment will work. On the contrary, we meet every kind of failure, which can easily drive us crazy. So it's more important, compared with working hard, to avoid bad influence from failure. Just relax your mind, lower the expectation, but never stop working hard.
And if playing violin and working out is a way to do that, spend that time. It's a great investment.
I mean, this was such a cool concept, and with the opening of the UN earlier this month, and the discussion on global climate change and renewable energy sources, and the Congress debating infrastructure, I thought, what better topic than to consider of how microbes can help us all out.
Michael, I think it's really important to emphasize that people are working on novel approaches to making energy, because we just came from an administration who said the problem is nothing, we just burn more coal. Remember that?
Yes.
And I just want people to realize that's not the solution. And look at Colombia. They're saying that's not the solution. And we have alternatives now. And, sometimes it costs a little more money for the alternatives, but in the long run, they're better than just, don't make it a political issue just like healthcare. It's crazy.
Well, let the science lead the way.
Charleston, to deal with global climate change, Charleston is building a seawall around the peninsula to keep the ocean out. And, we're spending billions of dollars to keep the sea out. I am certain that New York City is going to do something similar.
Sure.
If they have not already started. And
that water's got to go somewhere.
Yeah, the three of us, Elio, Vincent, and I, are in waterfront communities. Michele is in landlocked Michigan, though. Lake Michigan could always overflow and come visit Ann Arbor.
No
or
salt.
Lake Huron.
No salt in the Great Lakes.
That's good. No corrosion.
No corrosion. Well, there's corrosion. Where there's water, there's always corrosion.
Well, it's worse with salt, right?
Oh, yes. My
just
air conditioning systems are living proof.
Yeah, it just ruins everything. All right. On that optimistic note, that is TWiM 252, show notes, microbe.tv/TWiM. You can send your questions and comments to twim@microbe.tv. And if you like our work, consider supporting us. We'd love your financial support. Go to microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thanks, Michele.
Yeah, happy podcast day.
Happy podcast day by podcasting. Elio Schechter is at Small Things Considered. Thank you, Elio.
My pleasure.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.